In this video, in our last video of this unit, we're going to be working with the second cell process to move substances across the cell membrane, but this video is going to be all about active transport. So if you remember, kind of looking here, passive transport used no energy, okay, and it also moved substances from high amounts to low amounts, okay, so from high to low, okay, um, but in active transport, which is what this video is going to be looking at, we're going to be looking at, or it's only working with, I should say, movement of materials across that cell membrane with the use of energy. Now, since it is using energy, the reason for that is because we're moving materials or substances against that concentration gradient. So that means we're going to move these materials from a low concentration to a high concentration. Remember, concentrations can also be known as amounts. Now, what that means is active transport is going to need to use something. And so it uses what are called transport proteins. Now, we've worked up with proteins in the previous unit. Okay, we know what their structures are like. They're made up of amino acids. But transport proteins, they're going to actually change shape. And when they change shape, that's occurring simply because that's when they bind to the molecule that's going to be moved across this membrane. Now, these are often called pumps. So you might hear me say like protein pump. We're talking about transport proteins, so they have another name to them. And so how they're going to use this energy is they're going to break down a molecule known as ATP, okay, adenine triphosphate, to use this energy to pump the molecules across the membrane from the low concentration to the high concentration. So that's what this image that I started off this video with is showing you, okay? So we're going to be going from low amounts to high amounts from inside or outside of the cell, but we're using now this pump, but the pump requires energy, and energy is ATP. Now, an example of this type of pump, so I'm going to put an example here, would be when we need to move sodium, remember that's Na, and potassium, that's K. So it's called the sodium potassium pump. What it is it's where you have high amounts of this potassium ions. That's why you're going to see it with a charge. And you have low amounts of the sodium ion. And so what's going to happen is it's going to pump, let me go back to this picture, it's a little bit easier. It's going to pump out 3 sodium ions as it pumps in 2 potassium ions. And it keeps doing this back and forth, and it's pumping it from low concentration to high concentration while also using energy. Okay, now sometimes though, there are molecules that are a little too large to be transported. Because potassium and sodium, they're pretty small. So if they are too large, there's 2 separate processes to get these molecules across the cell membrane. And that first process is known as endocytosis. Endo meaning going into the cell. If we kind of break this up, okay, that's what that means, going into the cell. And so it's going to take materials in the cell, and it's going to it uses what is called a vesicle. Okay, and so this vesicle takes materials into the cell by simply engulfing. It looks like it's going to like open up its mouth and eat it. Okay, so let me kind of show you this before we write more. And so we're trying to get these large molecules into the cell, but it has to cross this membrane. So this is a vesicle. You can kind of see right here the vesicle starting to form. It like opens up its mouth is what it looks like, even though it's not, okay? And we are opening up, and then we're going to close around it, and then we're simply going to bring it in. So the process is the cell membrane makes a pocket. So here's your pocket, okay? Then the pocket's going to pinch together. So here's your pinching part around the material. Then you'll notice that in these two pictures, the vesicle is breaking off. And then it actually forms the whole vesicle inside the cell. And the vesicle is going to join a lysosome, okay? And then it's going to break down those contents for whatever their uses may be, okay? Now, there's also something called phagocytosis. So I'm going to write that word down. I think that's an interesting word. Plus, for my visual people, or you could say it here as phagocytosis. This is where instead of the lysosome engulfing the food, the cytoplasm will engulf the cell's food. Okay, so this is the whole idea is bringing large molecules across the membrane. Now there is a second type of process, like I had mentioned, and this second process is the opposite, exocytosis. Exo meaning to exit. So this part means to exit. So this is where we are trying to release these larger molecules outside the cell. And the process is just the opposite of what we wanted or what we just talked about. That's an F or from. So if we look at the process, I'm going to go to another picture. Okay, what's going to happen is the vesicle is going to form around. So remember, the circle thing is your vesicle. It's going to form around the material. It's going to move, so you can kind of see it's getting closer and closer to the cell membrane. And then as it gets close to the cell membrane, it's going to release. So here is it releasing the products to the outside of the cell. And so this is an example of like your digestive enzymes, or how we transmit nerve impulses, or even releasing of hormones from inside the cell to outside the cell. So we talked about this process, active transport, needing energy, right there, ATP, there's your energy to move across these molecules. But we also talked about moving larger molecules across this membrane, either in or out of the cell. And so those two processes are known as endo or exocytosis.